So now we're going to move on to identification of important genes. And the whole idea here is that when we do single-cell RNA-seq, we get expression for usually 10,000 or so different genes. Most of those are not going to be responding to whatever biological conditions we're looking at. It's usually only a few thousand are actually responding strongly. And if we can get rid of all the genes that are not responding to the biological condition, then our clustering or pseudo-time or differential expression methods will be much cleaner without all the noise from genes that are just subject to technical noise, rather than having real biological differences. It could be used for bulk RNA-seq as well, although most of the methods people use are not applicable for bulk RNA-seq because you need a large sample size to get reliable estimates of the different values they use. But the idea could be applied, yes. So for our method, we're going to be looking at the dropouts because the key difference between single-cell RNA-seq and bulk RNA-seq is that there's a large number of dropouts in it. And we're going to be using this data set called Tzoskin, which is a bunch of mouse neurons. And you can see the different neuron types that are present. So there's 4 different neuron types in this data set. So we're going to look first, we have to normalize the data, which I've got this nice function to do in the package. And then we fit a function. In this case, it's the Michaelis-Menten function. So this is the Michaelis-Menten constant. And this is the mean expression of the gene. And when you plot this, you'll see that there's this very tight relationship between the dropout rate and the mean expression on the x-axis. And you see that the Michaelis-Menten in black fits this relationship quite well. You can also use a logistic function in purple, which fits almost equally well. There's been other models proposed, such as this one, which don't actually fit the data very well in most cases. So the idea we're going to use to identify biologically responding genes from this is to look at the right outliers. Because if we have a gene that's highly expressed in one population of our cells, it will have a low dropout rate in that population and a high mean expression. And if it's lowly expressed in a different subpopulation within our data set, it will have a high dropout rate and a low expression for that. And because there's a nonlinear relationship between that, when we average across these two populations, we'll end up somewhere shifted to the right of the curve. So those differentially expressed genes should all end up to the right of the curve. So here we've just devised a method called M2-drop differential expression, which tests each gene to see whether they're significantly shifted to the right of the fitted curve or not, keeping track of all the different errors, so the error in mean expression, the error in dropout rate, and the error of our fitted curve. And you can see that there's a large number of genes that are detected using that method. So if we take a bunch of known neuron markers, we can check whether this method has identified them as differentially expressed. And you can see that almost all of them have been detected as differentially expressed using this method. Another way of checking whether these genes really are differentially expressed is simply to plot the expression values. So I've got this heat map function. And you can see that indeed most of the genes appear to be differentially expressed across the different cell populations. So here you can see a whole bunch of genes that are just expressed in the NP neurons, whereas over here we've got genes that are just expressed in a subpopulation of the NF neurons. And so we don't need to know what the groups are ahead of time, so we can use this as sort of predict how many clusters we might want for our clustering method. or to, based on this, tell whether we've got strong clustering or whether we would prefer to have some sort of pseudo-time analysis because the gene expression is more continuous across the cells. So we can now compare this to other possible methods. So one alternative way to do this is to use differential expression. So if you know your different cell types, you can use standard differential expression methods to rank the genes based on how differentially expressed they are across the known cell populations, which I've already done, and you can load using that function. And the alternative to using dropouts is you can use the relationship between variance and mean expression, which if you see plotted here is a lot more noisy than the relationship between dropouts and mean expression, but it's commonly used as well. And similar to the dropouts, you're just looking for genes that have higher variance compared to the rest of the genes in the data set as being highly variable. If you have spike-ins in your data set, you can fit the curve just to the spike-ins as your null model of technical noise, and then find those genes that are more variable than the spike-ins. But if you do that, you'll often find that the spike-ins over here at the high expression level will actually be above almost all of your endogenous genes. So you generally won't detect genes that are differentially expressed that are highly expressed as well using this method. So as an exercise to plot the heat map for the highly variable genes to see if they look differentially expressed and to see how many markers are identified using that method. And then we can compare the genes identified using differential expression versus highly variable genes and differentially high dropout rate genes. And you can see that N3 drop is pretty conservative, whereas the two other methods find a lot more genes, though they're not very consistent as to what genes they find. So I'll give you a few minutes to work through the exercises on this page, which is mostly just comparing the highly variable genes to high dropout rate genes. And then we will move on to pseudo time. Okay, we're going to move on to pseudo time now. So if you did the exercise, you should find that detecting highly variable genes, you get a lot of genes that are lowly expressed in almost all the cells and are just sort of randomly highly expressed in your data set, which you don't find when you use the high dropout rate method. But you'll still find the markers. So now we're going to move on to pseudo time, which is sort of the opposite idea of clustering. So when you're clustering, you think you have very distinct cell populations that are all very different from each other and relatively homogeneous within them. When we're thinking about pseudo time, now we think we have sort of a continuum of cells that are starting in one state and then gradually changing into one or multiple final states. So we expect a much smoother continuum rather than distinct cell populations. So it's used a lot if you're looking at time series across cells, or if you're looking at a differentiation process, or it could also be used for if you have cells that are located on some sort of chemical gradient to align those cells along that continuum. And the reason we have to use pseudotime rather than just using the sampling time point is that cells don't change and respond to signals at the same speed. So some cells in, say, your third time point will have finished their differentiation, whereas some cells will only be halfway through, and some may not have differentiated very much at all. And so comparing the snapshot of the cell population at each time point can be very difficult and very noisy, because you have cells that are actually at different stages in the developmental progression at the same time point. So here the goal is to align the cells based on how far they're making it through that process, as opposed to what actual time point you saw them at. And there are many, many methods to do this. So this is just some of them. This is not an up-to-date list. Almost every month or every 2 months, there's a new method published or made available for working on this. So this is some of the most common ones, and you see that many of them are implemented in different languages and different places where you can find them. We're mostly going to be working on, we're going to be looking at Slicer, which is an R package, and Monocle and Tscan, because they're available in R. And at least Monocle and Tscan are quite robust because they've been submitted to Bioconductor, so there's some degree of error checking going on, whereas some of the others have been less thoroughly error checked. So this is a summary of all of the different methods. Essentially, what all of them are doing is you take your single cell expression data, you find some sort of similarity between the cells, you learn some sort of path or trajectory within that matrix of similarities. You may perform clustering on it as well, and or create some sort of graph linking all of the cells together based on their similarity, and then using some sort of path-finding algorithm, try and find a route from one end of that network to the other end. And you can see there's lots of different variants you can use. So there's lots of different ways to find some sort of dimensionality reduction or learn some sort of path in that data. There's several different ways of clustering. There's many different ways of defining a network amongst those cells. And then a few different algorithms for looking at different paths within that network. And then lots of different ways to use that path to project all of the cells onto that path in some sort of order. The methods also have different pros and cons. So some of them, like Tscan, are very fast and can be used for large data sets, whereas others, such as Monocle, are not, and you have to shrink your data set down before you can use it, or it's only applicable to small data sets. The different ease of use as well. Some of them have very good documentation, some of them don't. And they can have different biases as well in there. So when you're doing this, try and find a method that is applicable to your particular problem. So the first method we're going to look at is Tscan, which is a method that uses clustering as well as networks for pseudotime analysis. So it's based on a mixture of normal distributions, which it uses to find clusters in the data, and then builds a tree connecting the center of those clusters, and adds branches if necessary. So if you load this data, load this data and run the method, this data set is mouse development embryos. So it's from the zygote up to the blastocyst developmental pathway. Your Tscan is not loading. That's weird. They're not loading.
Unable to load shared objects with our local library library tool.
That's clustering. Where is pseudotime?
Oh yeah, you need to, if you restart your R session, restart the R session. Session on top. Restart.
Restart. Yeah, don't save anything.
Run. Yeah.
Yeah, there it goes.
Yeah, so if you have a problem with Destiny, restart your R session, not Studio, and run it again. Clean R Studio.
Yep. Yeah. Restart. Yeah, restart the R session. Don't save anything, and then rerun it, and it should load correctly. So is anyone having difficulty getting this plot now? No more errors? Okay, good. So in this plot, you can see that in this command, we said we expect there to be 10 clusters. And this is based on the fact that there were 10 different sampling time points in this data set. So you can see each cluster here is in a different color and is numbered. And then these lines indicate the path that T-scan has found between these different clusters to put them in an order. And it hasn't found any branches in this case. So it's just one path, starting at 5 and going all the way to 4. So then you can use these commands to find an actual pseudotime. So here T-scan will project each cell in each cluster onto that path to define a particular pseudotime value for that cell. And you can check which actual sampling time points have been assigned to each cluster using that command. So here we can see that cluster number 10 has lots of 2-cell stage in it. So this is early on in the development. Whereas if you were to change this number 10 to, say, 6, you should find that it's lots of blastocysts, so later in the developmental time point. And then using these commands, we'll plot the cells based on their pseudotime and color them based on the actual sampling time point. So here, red is early and purple is late sampling time point, and you see the pseudotime fits this quite well until you get to sort of the blastocysts. These are all different blastocyst time points. And as well, the 8 and 16 doesn't differentiate very well. So we have an exercise here to try this with different cluster numbers. So if we go back up here, we can see that many of the clusters in here are kind of all overlapping. So you might want to reduce the number of clusters for this particular data set to see if you get a more reasonable trajectory or more meaningful clusters. Yes. Yeah, so the question is how you would select the number of clusters. And yes, you can use methods like we showed before in the clustering section to decide how many clusters would be appropriate for your data set. So any questions on T-scan? Nope. Okay, so the next method we'll look at is Monocle, which is quite similar to T-scan, only it doesn't perform clustering like T-scan does. And it unfortunately does not run well when you include all of the genes. You have to select a smaller number of genes before running it. And it crashes RStudio. So if you're running in RStudio, it is known to crash RStudio. So don't be surprised if it crashes. So we selected genes using M3 drop, but Monocle now provides its own feature selection method, which is extremely complicated, which is why we're not talking about it. And then to run Monocle, you have to put the data into this cell dataset format, and simply run. So this set ordering filter, this will select. So here we're telling it to use the M3 drop genes for its ordering. And then we first reduce dimension, which does using PCA similar methods. And then it will order cells according to pseudotime. And then it will plot this trajectory for you. So the new version of Monocle, you do not have to say how many branches you expect. It uses some very fancy machine learning in order to decide what the optimal number of branches are for your particular data set, and then distinguishes them appropriately. So I believe this is actually, so here it's decided there's 3 states, so this root state and then possibly a root state. So there's one state that's sort of all together there, and then one state here and one state going down that branch. And then here we're going to extract the pseudotime from Monocle and get the time point, and then we can color the pseudotime versus the real time points again. So you can see Monocle is quite similar to T-scan, only it's got it reversed. It's picked the wrong route in this case. So here it's starting with the blastocysts and going to the 2 cells, rather than from the 2 cells to the blastocysts, which is quite common for pseudotime methods. Pseudotime methods can't tell the difference between one end of your trajectory and the other end. So you may have to reverse your data set. I believe Monocle has an option to say, yes, reverse your ordering in order to get to be the right direction. Any questions on Monocle? This will produce branches, yes. So this data set should, in theory, have branches, but none of the methods actually detect them. So the blastocyst should diverge into 2 different cell types, but none of the methods so far have been able to detect that that we've tested. And so here it's basically finding one branch. T-scan found one branch, although I was really hopeful for T-scan, because if you go up to T-scan, you can see that these 2 clusters are almost next to each other. So really there should be a separate branch going that way to 6, and then that would have been correct. But because the method will just connect the nearest cluster center, because this cluster is slightly closer to that one than to that one, it doesn't find the branching point. But if this cluster had been sort of over there, this would have produced a branch.
Would that tell us that 2
So the third method we're going to look at is diffusion maps, which is really an alternative to PCA, where it assumes some sort of diffusion process is going on that connects all of the cells to each other cell, and then infers a low-dimensional manifold that represents the sort of diffusion from one cell to the next cell. So this is how you run diffusion maps. So here we're choosing diffusion component number 1 and 2, but you can have more components than that if 2 components don't completely represent your data set. Which I believe is exercise 2, is to consider that. And so here we're showing that these early cells are much, much different, more different from the other cells than anything else. And you can't really tell the difference between the late time points at all. And Destiny is fast enough to be run on all genes, but you may want to perform feature selection anyway in order to clean up your data set and get better results. Questions about Destiny? No. Okay, so the third method we're going to look at is Slicer, which again, pretty similar to Monocle and T-scan, but uses different underlying statistical models. So Slicer, again, requires you to select the genes from the data set in order to model the data, rather than using all of the genes like Destiny did. And then in order to do its nearest network, you need to select a K. So this is essentially how many cells, nearest cells, do you connect each other cell to in order to build the network to connect all of the cells together. And you have to tell a K-min and a K-max in order for it to search all the different Ks to select the best one for you. So here we've done 30 and 60 because it gives quite good values. And then here this command is projecting it down, projecting that network down into lower dimensions. And then we can plot it, and then coloring again by time point. So here we can see a nice trajectory coming out. You can also plot the k-nearest neighbor graph that it created. So here you can kind of see in 3 dimensions, this would be a single sort of spiraling network. that it's produced with no branches. And then once it's reduced the dimensionality down to this trajectory, this manifold, it will automatically find the extreme cells. So these should be the starts and potential start and ends for your data set. Although if you can see that when we apply it to this data set, it actually picks the wrong points. So it's picked 2 points in the middle of our trajectory, not the 2 endpoints. And then it allows you to define which of those extreme points should be the start of this pseudo time versus the ends. And then it will calculate the pseudo time for you and assign the cells to different branches, if there are multiple branches, which there aren't in this case. And then because it's got the wrong start point when we plot the pseudo time, we can see that it's got these 2 different parts in the wrong order because it picked the wrong starting points. So similar to t-scan, because you have this input parameter of k, you can get very different results depending on what k you use. So here I recommend you try k equals 5 and see what happens when you use that. Okay, hopefully all of you have got a chance to try for yourself that if you change the k here, so that k to 5, you should see a plot with only a couple of points on it, each a different color, because essentially what's happened is that all the variation within each time point has been eliminated, and you're just capturing the differences between the time points. So each point, each red dot is overlaid on every other red dot, every red cell in there. So it's very important to choose the right K for this method. And we're going to break for lunch. So come back around at 1:30 and we'll do differential expression for single cell data sets.